Hi guys. Today we will be going over chapter 3, which covers cells. Um, Kind of an overview of cells. So different size and shape of cells are found in the human body. They're going to vary in size depending on type, but all will be microscopic, so you won't be able to see them with the naked eye. Um, the biggest way that they differ is through shape, though. So shapes can be flat, round, donut shaped. It's going to depend on the specific purpose of that cell as to the shape that it takes. If you think about our red blood cells, they kind of have that donut shape where they're thicker around the edges and then kind of flat in the middle. And it just so happens that an oxygen cell or molecule sits right on top of that little donut shape perfectly. So that's an example of how shape meets function. All of our cells are going to be composed of three different areas. So we have the plasma membrane, which is a thin membrane surrounding the cell body, kind of forms an outer boundary. What it does is it keeps the stuff that's supposed to be inside the cell, keeps it in there, and anything that's not supposed to be in it or that will be harmful to the cell, it acts as that barrier to keep it outside of the cell. Composed of a thin two-layered membrane of phospholipids. Another thing that's important to note about this plasma membrane is that they are selectively permeable. So what that means is that depending upon the cell type, certain materials are allowed to enter and exit the cell body, while others are kept outside. So that plasma membrane, you can kind of think of it as the bouncer at a club. They're just going to let the people that they want in or the matter that they want in, while keeping others out. Anything that could be damaging, it's keeping out. The next area is going to be the cytoplasm. So this consists of all of the living material inside the cell, except for the nucleus. The nucleus is going to be kind of its own thing that we'll talk about here in a second. Within the cytoplasm is a component called the cytoskeleton, and what this does is provide support to the cell, very similar to the way that our skeleton or the bones in our body provide support to us. And then the last area is going to be the nucleus, which is a large structure inside the cell that contains the genetic code of the cell. So that's kind of the brain of the cell, if we're continuing that comparison of a cell to a human. Cellular organelles, so these are going to be found within the cytoplasm. I'm going to just briefly touch base on these, and then we're going to get more into cell transportation processes, and mitosis is really what we'll focus on during this lecture. Of course, look over these slides and read about the organelles in your book, but I'm just not going to spend a lot of time lecturing on them. Most of the cells within the cellular organs within the cell are going to be focusing on the production and transportation of proteins. So proteins are required for the structure, function, and regulation of the cell, and ultimately the tissues and organs, because they are composed of those cells that we're talking about. So just remember to relate everything back to those proteins. So ribosomes are going to manufacture enzymes and other proteins, commonly referred to as protein factories. The endoplasmic reticulum is broken up into rough and smooth. So rough is responsible for collecting, folding, and transporting proteins, which were made by the ribosomes. And then smooth endoplasmic reticulum is responsible for synthesizing lipids and certain carbohydrates. The Golgi apparatus is a group of flattened cells near the nucleus that collects chemicals into vesicles that move outward to the plasma membrane. So that's kind of a transportation center. They call it the Chemical Processing and Packaging Center. Mitochondria are involved in chemical reactions that result in the release of energy. So they are often called the power plants of the cell. They're responsible for the energy of the cell. Lysosomes are membrane-enclosed packets which contain digestive enzymes. So lysosomes are responsible for a process called hydrolysis, in which large molecules are broken down and broken apart. So for this reason, the lysosomes are sometimes called digestive bags. And then the last organelle that we'll talk about is a centrosome. So this is responsible for organizing the microtubules of the cytoskeleton. So again, that cytoskeleton is what gives the cell its support and its shape. It does also play a role, the centrioles play a role in organizing and moving chromosomes during cell reproduction, which we'll talk about here in a little bit. Some cell extensions, there's 3 different types. So microvilli are small and finger-like, and their role is to increase absorption. So the area that we're most likely going to find these is somewhere like the small intestine, where we're trying to absorb those nutrients from the substances that are passing through. So the microvilli are responsible for absorption. The cilia are fine hair-like projections, and they allow movement. So when we think of cilia, we think of the respiratory tract in the throat, helping to move that mucus up. So those cilia kind of move in a wave-like fashion, so kind of projecting the mucus up the throat or the respiratory tract. And then the last one is the flagellum, and this is just a single projection that comes from the cell. Excuse me, its main purpose is to enable movement. So there's really only one cell in the human body that has this extension, and that's a sperm cell. So the little tail that comes off of the sperm is actually a flagellum or flagella, and it allows the movement of that sperm through the male reproductive tract and then potentially into the female reproductive tract. So the nucleus is going to contain our genetic code. So that's responsible for controlling the organelles in the cytoplasm. This is what kind of gives the cell its purpose. It kind of directs the cell to where it needs to go. Some structures that you'll find in the nucleus include the nuclear envelope, which is an outer membrane composed of 2 separate semi-permeable membranes. So if you think of the nuclear envelope and the plasma membrane, it's kind of the same thing, but the plasma membrane is surrounding the whole cell, while the nuclear envelope is just surrounding the nucleus. Then we have the nucleoplasm, which is going to be similar to that cytoplasm. It's just cell material that contains the nucleolus and chromatin granules. So the nucleolus is nuclear material, so inside the nucleus, where ribosomal subunits are formed, and those are important in the process of genetic information. Here we'll look at the relationship of the cell structure and function. So we kind of touched base on this already. Every cell in the human body has a designated function. Some are going to help maintain the cell, others help us regulate our life processes. If we look at that sperm cell again, that movement of the flagellum is going to be an example of how an organelle has a particular function or how that cell has a particular function. So now we're going to start looking at that movement of substances through the cell membranes, specifically that plasma membrane. It can be broken up into 2 different types. So we have the passive transport processes and active transport processes. So we'll touch base on passive first. So passive transport does not require any added energy. So no energy expenditure is occurring within the cell for these passive transport processes to take place. You'll read it in your book. They refer to it as moving down a concentration gradient. So if you think of a ball rolling down a hill, gravity's kind of taking over. No energy has to be expelled for that ball to keep rolling. So that's what's happening through our passive transport process. So the first type is diffusion. This is where substances scatter evenly throughout an available space. The best example of this is a tea bag placed in hot water. So once you get that tea bag placed in, you can see how the tea evenly disperses throughout the water that it was placed in. In order for this process to take place, the membrane must have pores big enough for the particles to pass through. That component is really important. Osmosis is going to be the diffusion of water. So this occurs when solutes cannot cross the plasma membrane. Instead of the dissolved substances passing through that plasma membrane, the water will pass through to try to equalize or balance those water levels. Dialysis is the diffusion of small solute particles. So some will move across the membrane while others do not. This often results in an uneven distribution of those solutes. And then filtration is the movement of water and solutes caused by hydrostatic pressure on one side of a membrane. So that pressure is going to be the force or weight of fluid pushing against a surface. So this occurs mostly during urine formation. So this just shows diffusion through a membrane. So you can see here, when we start, we're looking at 10% on one side and 20% on another. So what happens is both solutes and H2O diffuse through that membrane. So for the side that has 20% more, we're going to move some solutes over to the 10% side, resulting in that even distribution that we'll see here. That's a really bad circle, sorry guys, but that's the even distribution that we're looking at. So now we'll look at active transport processes. The most common one that you're going to see is ion pumps. So what happens is there's an actual pump that sits in that cell membrane, in that phospholipid layer. And what happens is energy from ATP is going to cause the pump to move substances across the cell membranes against their concentration gradients. So we talked about passive processes, how that ball is just kind of rolling on its own, nothing has to be done. With these active transport processes, we're going against the concentration gradient, so we're ultimately going uphill. So if you think of a person trying to push a ball uphill, there is going to be energy required to continue that process. So that's what's occurring during that active transport process. One thing to note is that ion pumps are specific. So a sodium pump will move sodium ions, calcium pumps will move calcium ions. There are sodium-potassium pumps that will move sodium and potassium, but just know that if a calcium ion tries to get into a sodium pump, it's not going to do anything. That process won't work. So the next active transport process that we have is phagocytosis. So this refers to cell eating. So this is when a cell is engulfed in a vesicule as a protective mechanism. Where we're going to see this is in the destruction of bacteria. or debris that might get into the body through tissue damage. Penocytosis is cell drinking. So this is going to occur when fluids or dissolved substances are engulfed. So keep in mind, phagocytosis is particles, and penocytosis is fluids or dissolved substances. So that's the big difference between those two. Here is your picture that's in your book of that sodium-potassium pump. So this is an ion pump. If we start here, we're looking at the extracellular. So that means we're outside the cell. Intracellular is going to be inside the cell. So we'll think about those medical terminology components as we're working through this. So we have our sodium ions here that are trying to get out of the cell. So they're going to go into the pump right here. ATP allows that energy to be released, and the sodium will be pumped extracellularly. So they'll move their way out of the cell. At the same time, we have potassium that comes in from extracellular, and it is again going to just travel through that pump into the intracellular. So now we're going to talk about cell growth and kind of the processes that are involved in cell reproduction. What we have is deoxyribonucleic acid, or your DNA structure. This is going to contain the chromosomes. The chromosomes of a cell are what give us our features. They help guide the direction of the cell, that sort of thing. So the DNA structure is a large molecule shaped like a spiral staircase. Double helix is that term, that kind of spiraling staircase. Within the DNA molecules are genes, which is our genetic code. This is going to determine, like I kind of mentioned before, the structure and function of the cell and the heredity. So when you hear of certain features being passed down from generation to generation, those are in our genes. The genes are responsible for those hereditary characteristics. All right, so starting kind of in this section is where things can get a little bit confusing. Keep in mind, if you guys need any clarification on this or any outside resources, anything like that, please just let me know, and we can figure out a way to kind of look at the information from a different point of view to help really cement these ideas. So we'll start with nucleotides. So this is going to be a smaller unit of DNA. So if we go back here, we can kind of see in this picture that there's different little units of those DNA, or those different colored sections. These nucleotides are composed of a sugar, a phosphate, and a base. So the base pairs, or the bases, excuse me, are going to be adenine, thymine, guanine, and cytosine. These base pairs are complementary. So that means that the same two base pairs always pair with each other. So adenine and thymine are going to pair, and cytosine and guanine are going to pair. So whenever you look at those DNA molecules, you'll see that there's kind of a split down the middle. Typically it'll be one color on one side, another color on the other. That's demonstrating that complementary base pair. So one side would be adenine, the other side would be thymine, or it could be cytosine or guanine. Just keep in mind that those base pairs are always going to be the same. Another thing to note is that just because the base pairs are the same, the sequence is not. So it won't always be adenine, cytosine, cytosine, adenine. Those can vary, and they will. That's what gives us the genetic code, and kind of acts as our indicator as to which characteristics we will develop. So next we have ribonucleic acid, or RNA. This is going to play a role in protein synthesis. RNA is going to be single-stranded, where if we think back to our DNA, that one is double-stranded. So its main role is to copy information from the nucleus and carry it to the cytoplasm. Once it's in the cytoplasm, we're going to begin the process of protein synthesis. So this is the transferring of genetic information from the nucleus to the cytoplasm, where the proteins are produced. So the nucleus is kind of the driving force behind the cell. So what it's doing is sending out a message to the cytoplasm. So that genetic information is leaving the nucleus, going into the cytoplasm, where those organelles are going to begin the process of producing whatever specified proteins the nucleus has said, packaging, and then putting them out of the cell into the body. To do this, we're going to have to go through transcription and translation. These two processes can be very, very confusing. I'm not going to spend a lot of time on them. Honestly, it's important to know that these two processes are responsible for protein synthesis, but really digging down into the actual processes tends to lead to just confusion and not really an understanding of what's really occurring. So don't get too caught up on these. If you have questions, for sure, let me know. But I'm not going to spend a lot of time on them. So transcription is where the double-stranded DNA molecule separates to form messenger RNA, or mRNA. So these are going to pass from the nucleus to the cytoplasm, where they begin to direct protein synthesis in the ribosomes and endoplasmic reticulum. During this transcription phase, each strand of mRNA duplicates a particular gene from a segment of DNA. So it's basically, I think of them kind of as carbon copies of that DNA. And then that is what goes out into the cytoplasm and kind of directs those organelles into the direction that they need to go. Once that information is in the cytoplasm, we can begin translation. So the information contained in the mRNA is in the cytoplasm now. The ribosomes are going to begin that process of protein synthesis. So they've read the information on that messenger RNA, and now they're beginning the production of those proteins, which, if we think back, are responsible for the actions of the cell. I'm not going to go into a whole lot more detail with that. If you guys have questions, let me know. I have a couple of videos that I can share with you as well that will hopefully kind of demonstrate these processes. So now we'll talk about cell reproduction. So during reproduction, the nucleus is going to divide, and this is a process called mitosis. During that time, the cytoplasm is also dividing as well. So once this division has taken place, we have two daughter cells, which are the exact copies of the original cell. So we'll start with interphase. This is going to be the period when the cell is not actively dividing. This is an initial growing stage of new cells that contains a little period when the cell is preparing for cell division. Then we're going to get into mitosis. So this is the actual division of the DNA in the cell, broken up into 5 different stages. So we'll start with prophase, which is the first stage. During this time, the centrioles are moving away from the nucleus. That nuclear envelope disappears. So there's no membrane holding back the nucleus, and that genetic material kind of floats throughout the cell then. Another thing that occurs is the spindle fibers appear. So these are kind of the guide wires that assist chromosomes in the movement towards the opposite ends of the cell that we'll see later on in mitosis. Metaphase is your second stage. During this time, the chromosomes align across the center of the cell, and the spindle fibers are going to attach to each chromatid. So there's some pictures in your book that kind of demonstrate this if you want to kind of follow along while you're listening to this lecture. Anaphase is the third stage. At this point, the chromosomes are pulled to opposite ends of the cell, and a cleavage furrow develops at the end of this phase. And that's just kind of the beginning of that separation. There's kind of an indentation in the middle of the cell. Telophase is the fourth stage. The nuclei begin to appear in the daughter cells, and the nuclear envelope and nucleoli appear in those cells as well. So we end up with 2 nuclear envelopes, 2 nucleoli, 2 nuclei, basically 2 of everything, 1 in each of those daughter cells. The cytoplasm is then divided and the daughter cells become fully functional. So when they say that mitosis is a 5-stage process, they're actually including interphase into that component. So they would say interphase is step number 1, then we'll go into prophase, metaphase, anaphase, and telophase. A phrase that people use to remember this is I passed my anatomy test. So I, interphase, P, prophase, M, metaphase, A, anaphase, and T, telophase. Here's that diagram that's also found in your book, just giving you a visual of how the cell will look during those different processes. So once cell division is done, we'll have 2 identical cells. One thing that can occur is differentiation, and this is a process where the daughter cells actually specialize and form different kinds of tissue. So this normally occurs during times of body growth, so more in that puberty stage, or even in infancy when when the body is just beginning to grow and develop those different kinds of tissues. If there's abnormalities during these division processes, neoplasms, which are tumors, can occur. So those can either be benign or malignant. Malignant will result in cancer. So this is kind of where that beginning process of cancer cells occurs. So that is chapter 3. This chapter is one of the harder ones that we'll cover. There's just a lot of components in it, so please don't hesitate to contact me if you have any questions or just need any clarification on any of the topics. Thanks.